Wow. Well, you just had lunch, right? So nobody's going to be sleeping after that. I'm so pleased to meet you. My name is Mark Petrash. I'm the Vice Chair for Research in the Department of Ophthalmology. And John had invited me to come and talk a bit about our research program here. And I'm really happy to do it. Catch my breath after that. That's you. Let me just say what a pleasure it is for me to do this because I see my father in you. I grew up in Baytown, Texas. My father was a school teacher there, and he was a lion. When I was in college, in fact, when I was studying doing my doctoral research, he asked me to come and talk to his Lions group about my research. And this was in Baytown, and I just remember how proud I was to go and meet his club. We probably did in the warm-up. But, it was inspirational to me because he had reached his retirement years, and he wanted to stay involved in the community. And this was one thing that he did, was to join the Lions Club. And I just remember how meaningful it was to him. And I can remember how proud I was to go to his club and talk about my research. And I'm not going to say how many years ago that was, but let me just say it's been quite a number of years, and I'm really proud to be here to repeat that process. At that time, I was a student. I had my whole education ahead of me, the rest of my training. The early part of my career was yet to be established. And now I can look back at that and think back about what an excitement it was for me to share my research with that group and to see how proud he was of me and how the Lions could be part of that is a really great memory for me. So as I mentioned, I'm from Texas. I was born in Baytown, Texas, a suburb of Houston. Went to the University of Texas at Austin, where I studied science. And then after that, I did my doctoral research in Galveston, Texas, at the University of Texas Medical Branch, where I was under the training of a brilliant scientist who got me interested in inherited eye disease. At that time, we didn't know much about the genetics of eye disease. We were just beginning to understand what DNA and how genes were transmitted from generation to generation. We knew that some of these eye diseases could be handed down through the generations, but we had no idea what the mechanism was. And so that was my passion. And when I did my doctoral work, was to understand how genes work, how they're structured, and how changes in the gene structure gave rise to inherited eye diseases. And I have really been working on inherited eye diseases ever since that time. So it's really a great beginning for me. After my doctoral research at Texas, I went to New York University. Big change, big cultural change from Baytown to New York City. Spent 3 years there, and then had the opportunity to start my faculty career at Emory University at the Emory Eye Center, which at that time was one of the up-and-coming places. At that time, it was what University of Colorado was when I came here in 2008. Very good leadership, a commitment to research, a commitment to highest quality eye care, and a desire to build a world-class research program. And so I was lucky enough to be recruited there as an assistant professor. I was there for almost 4 years, and then got recruited to St. Louis, to Washington University, which was, again, had a new chairman, a new vision for what they wanted to accomplish with research. And I was there for 20 years and went through all the faculty ranks from assistant professor all the way to full professor. I was the director of research in that department, built a very large, very strong research program. And then in 2008, was recruited to come to Colorado. And my personal story on that decision was that I had, at that time, been involved in our research for almost 25 years. And we had developed the technology to be able to create new products that we wanted to bring to the clinic. But Washington University just did not have the infrastructure for that. They were really focused on basic research, but not translating that research into a clinical product. And it was very frustrating for me because as a scientist, I wanted to do more than basic research. I wanted to create something that would have an impact, and an impact to patients who were suffering from eye disease. And what I learned was at the University of Colorado, that was in their DNA. That was part of their genetics. They wanted to be able to sponsor research that would come out of the laboratory into the clinic. So it was a really easy decision for me to make, to leave St. Louis, to come to Denver to this department. So the leadership of Dr. Mandava, this is a very high priority of his, is to translate research into clinically high impact therapeutics. And that's what brought me here. But when I came here, there wasn't much basic research going on. There was one other scientist. And I came from a department that had 20 scientists. And so my vision in coming here was to build up again a research program as we have established in St. Louis and create one in Colorado that would have that same strength at the scientific level, but in addition have the commitment to translational research and have the commitment to use basic research as a way of creating things that can be brought to the clinic. So that's what I want to talk to you about today and what we've done along those lines. I just also want to, I can summarize sort of what my goals were and what the goals Dr. Mandava and I worked on when I first came here was to accelerate bench to bedside translation of basic research discoveries. I wanted to establish a center of excellence in regenerative medicine, and this is something we're going to get to a little bit later. It's something you're going to have the opportunity to see for yourself the state-of-the-art regenerative medicine research that's being done in the eye. In 2008, when I made the decision to come here, some very dramatic discoveries have been made at other laboratories around the world to show that it's possible to take cells from an adult and to convert those cells into an embryonic state. And then to treat these embryonic-like cells in a way that those cells could then be transformed into specialized tissue. And the significance of this discovery was that it was no longer necessary to use fetal cells or embryonic cells that have these stem cell-like characteristics, cells that can be induced to develop into specialized tissues. It became possible to not have to rely on embryonic or fetal cells, but to use adult cells. And not only that, but let's say if you had a patient who had a degenerative disorder, who needed some kind of cell replacement, if they received cells from another donor, there would always be the possibility of tissue rejection. But if you could use that patient's own cells and develop that therapeutic tissue from their own cells, then you could avoid the problem with tissue rejection. All of this became possible in the mid-2000s. So in 2008, when I was considering coming here, one of my goals was to establish a regenerative medicine center for ocular diseases, taking advantage of these new stem cell therapies. And that's something that you're going to be seeing today, is the fruits of that labor. One of the other goals that I had was to establish an environment for training our next generation of scientists. This has become a really critical issue. And that is that we're not producing enough highly trained scientists to feed the machine. We have unfilled jobs in the laboratory because we can't find people who have the proper education and the training to fill those. And so it's really up to us to establish that pipeline. and for us to play our role in establishing a way to replenish this next generation of scientists. And then finally, one of our goals was to establish team science and collaboration between basic scientists and clinicians. These are really two different cultures. We love each other, but we speak different languages. And one of the challenges in a major medical center like this is to find ways to get clinicians and basic scientists on the same page and to work together to commit to each other's success so that together they can create much more than they can create individually. And it needs leadership, and it needs an environment that supports these kinds of collaborations. All of us work really hard, and to be successful, you can't be successful always alone. You need to collaborate. And part of the collaboration is sharing the benefits of what you do, whether it's financial, whether it's reputation, whatever it takes. And so a culture needs to be established where people are willing to share the benefits of the research that they do together and not just hold it for themselves. And so that's one of the things we really work on a lot. Within ophthalmology, there are major issues that we wanted to address. And we have focused on the diseases that are the most challenging and the most prevalent in the world. Diabetic eye disease is one of diabetes is recognized as one of the leading causes of blindness. The Lions have a big program in diabetic retinopathy, and we're really focused in on that. Macular degeneration, also one of the leading causes of blindness among the elderly. This is a degenerative disease. As you can see, the macula is the part of the eye where the fine vision is located. And this is a big problem that we need to work on. Glaucoma is the condition where the pressure inside the eye is elevated. It's called ocular hypertension. And that elevated pressure kills the nerve cells that transmit the visual stimulus that comes through the eye, transmit that to the brain through the optic nerve. So there are nerves that do that, and this high pressure in the eye kills those nerves. And so we need to find ways of protecting those nerves through what's called a neuroprotection strategy. Cataracted lens disorders, the cataract is the leading cause of blindness throughout the world. So we're working on ways to either prevent or delay the onset of cataract. And then we have an effort on epidemiology. One of the areas that I'm going to talk about in a few minutes is to take advantage of data analytics, machine learning, artificial intelligence, kind of computer-driven ways of taking health data that are in major medical centers like this and mining that data to get new insights into eye diseases and to get new insights into what kinds of patients benefit from certain kinds of therapy. Not all patients respond to therapy in the same way. And so we have these deep data sets that can be used to gain insight into disease. Regenerative medicine is also something, and this is something that the cell site folks are going to be talking about. Let's see if I can pass something. Yeah. So what I want to do is just give you kind of an idea of who are the people and the faces that are working in these various areas. In our program in diabetic eye disease, here are 6 people who are working on different aspects of it. What you see here on this, I can use this. This is a picture of a retina of a diabetic patient. One of the problems that happens in diabetes is the blood vessels in the retina become fragile and they start bleeding. And this kills some of the cells in the retina. And so one of the goals that we have is to understand what causes the retinal bleeding, what can we do about that, and can we identify therapies that can prevent this from happening. And so we have Dr. Mandava here, who's a vitreoretinal surgeon, Dr. Matthias, Dr. Olson, and Dr. Smith. These are all vitreoretinal surgeons who treat diabetic patients and who work with the basic scientists to come up with areas of most of the highest relevance research. And
Dr. Nagaraj and I are basic scientists who collaborate with, so this is that team science I was talking about. We collaborate with our clinician colleagues to work together on drug discovery, ways that we can identify compounds that will slow down this bleeding process, trying to understand why elevated blood sugars give rise to retinal bleeding. So diabetics, one of the biggest problems they have is the high blood sugars. And we're trying to understand, why does high blood sugar cause retina to start bleeding? And if we understand that process, can we block it? Can we prevent it? And then inflammation is a really big area. And Dr. Smith is leading an effort to try to understand if inflammation gives rise to some of these retinal problems. Under macular degeneration, again, we have a strong team of retina clinicians, Dr. Mandava, Jeff Olson, Jesse Smith, and Mark Mathias, who see hundreds and hundreds of macular degeneration patients every year. Here's a picture of, another picture of a retina from a patient with macular degeneration. And what you see here is the accumulation of metabolic byproducts that accumulate in the retina of AMD patients and kill some of the light-sensitive cells in the retina. And this is what gives rise to blindness and macular degeneration. The teams are working together to conduct clinical trials of new AMD drugs that are being investigated to prevent this kind of lesions from forming in the retina. New methods of surgical intervention, and ways that we're going to be talking about later with using the regenerative medicine to be able to transplant stem cell-derived retina tissue into patients with macular degeneration. That's not ready for the clinic yet, but it's something that's very aggressively being pursued. In the glaucoma area, we have two clinicians, Dr. Kohuk, who I think maybe be known to you, and Dr. Pancheva, and then Dr. Nagaraj, who are working with new kinds of therapies for neuroprotection to protect the optic nerve. So this is the nerve that I was talking about that carry the signal from the eye to the brain that gets killed in glaucoma. What happens is glaucoma patients over time, and this is kind of a silent disease, so you can't really tell that you have glaucoma unless you go to the eye doctor and get tested. They have these visual field tests, and what happens is the cells die off in the eye, and they give rise to areas of your vision where you just can't see anything. So the visual field is obliterated progressively over time, and they're trying to figure out ways to prevent that from happening and to catch it early and prevent it from happening. One of the other issues with glaucoma patients is the glaucoma doctor, to relieve this eye pressure in the eye, will create a hole in the side of the eye that allows the fluid to ooze out. And over time, that hole can scar over and become fibrotic. And this is a problem because the patient has to come back in and has to undergo another operation to create one of these flaps where this pressure gets relieved. And this is called fibrosis. And so we have a really big program trying to understand what causes fibrosis after glaucoma surgery, and what can we do to prevent that from happening so the patient doesn't have to come back in for surgery. Cataract and lens disorders is another big area. This is one of my particular areas of interest. Again, me and Dr. Nagaraj, Dr. Davidson, and Dr. Christopher all work together to try to understand what causes the clouding of the eye. So this is a picture of a patient with cataract. You can see the very center of the eye. This is where the lens is located, and it turns white. And when it turns white, it prevents light from going through the lens and therefore can't reach the retina. And so we're trying to figure out ways to reverse this process. And so Dr. Nagaraj and I collaborate with Dr. Davidson and Christopher to come up with new drugs that can prevent this opacification process from happening. Coming up with ways that we can get these drugs into the lens. So the lens is like in the very center of the eye. And if you put an eye drop on the surface, how is that drug going to get from the surface of the eye all the way to the center? And we're coming up with ways to stimulate that process. And then finally, we Many cataract patients have to come back in for another procedure after about a year or so because they have what is called after cataract. And this is an inconvenience. There's some risk that's associated with this after cataract. We're working on ways to prevent that after cataract process from happening. We have some really exciting developments on that. The cell site program is our ocular regeneration program, our ocular stem cell regeneration program. This was the one of the things that I wanted to develop in coming to Colorado, and it was successful in recruiting what I would consider the world's best team to come from Johns Hopkins University in Baltimore to join us here in Denver. And there were 3 scientists that came, Dr. Cantal Solar, who many of you have met here as the head of our cell site program, Natalia Vergara, who's going to be giving a tour after my talk here, and her associate, Miguel Flores. These 3 scientists led a group in Baltimore who were able to establish for the first time a means of taking stem cells and growing retinas in a petri dish from these stem cells. And what was really miraculous about this is that these cells were able to form the specialized layers of the retina that you see in an adult retina. But this was done in isolation in a petri dish. And I think it's pretty miraculous, really. And not only that, but these cells, these retinas that were grown in the petri dish, were able to respond to light. And so what happens when the retina gets struck by light, it induces a chemical and electrical change in these cells. And this electrical change leads to a nerve impulse that goes to the brain through the optic nerve. And they were able to show, in the absence of a brain, in the absence of the central nervous system, these stem cell-derived tissues were able to respond to light electrically in the same way they would as if they were in the eye itself. And this then laid the foundation for the possibility of developing a tissue that could be transplanted into the eye of a patient with macular degeneration where those tissues had degenerated. And we know that when in the natural eye, when the tissues regenerate, they don't have the capability to replace themselves. They can't reproduce themselves. So the only way to restore the ability of the eye to respond to light would be to bring in new tissue. And we wanted to be able to bring in new tissue that wouldn't be rejected by the immune system. And that would be patient-derived stem cells used to grow a therapeutic retina. And so that's what CellSite is working on. And you're going to be able to see some of that this afternoon. Some of the cells. These are microscopic images of what are called retinal organoids. So these were the very early stages of these stem cells that have formed into mini retinas, and this is what they look like under the microscope. I think you're going to see some of those today. So CellSite, their major research goals is to develop a stem cell-derived retinal transplant, to develop the surgical methods for introducing this tissue into the eye. So it's one thing to grow it. It's another thing to grow it in a way that will be safe to put into the eye of a patient. And it's yet another way of how are you going to get it in there? It takes really specialized hardware and devices to be able to carry that tissue from the laboratory to the OR and then to introduce it into the retina, into the eye, in a way that would be able to allow that tissue to engraft into the eye. And so these are the areas that they're working on. These are really challenging areas. As I mentioned before, we have put a lot of effort into taking advantage of data processing and data analytics. A large academic medical center like this sees hundreds of thousands of patients a year. There are electronic medical records that are generated for each of those patients. The patients that come through the ophthalmology department all have electronic medical records. And it's possible for us to tap into those records and to look to see if there are patterns of response to certain kinds of surgical therapies, certain kinds of drug therapies that are offered to these patients. And it's a valuable resource that previously was going unused. And so we have invested in the creation of new programs to bring in specialists who know how to tap into that data and to make sense out of it. This is called an area called epidemiology. And so these are the three faculty members who we have who are focusing on this. Is Dr. Ann Lynch, Dr. Jennifer Betnaik, and Dr. Brandy Wagner. And what they do is to look for risk factors in these medical records that associate with eye disease, and they look for different aspects that can be used to predict surgical outcomes. They are also working with basic scientists to see if there are different molecules in the blood of patients who are being treated, and if these molecules can be used as what we call biomarkers. So if there's a certain blood protein, let's say, that's elevated in a patient with macular degeneration that responds to a certain kind of therapy, that would tell the doctor, like, okay, if we know that if that marker is elevated, then they should be treated with so-and-so therapy because we know it's more effective for a patient like that. So this is working really smart. This is using patient data and these medical databases to help guide therapy. You might call it individualized medicine. And it's a way of taking advantage of this information so you can provide the best outcome for care. One of the other areas that we've invested in is called Cochrane Eyes and Vision Program. This is a team of scientists, data scientists, who take advantage of all of the published medical literature and to be able to combine the outcome of studies done all over the world to identify treatment areas and certain kinds of therapies that seem to give the best outcome. And previously all of this data was out there, but no one ever put it all together to make sense out of it. And so this is a team of 3 data scientists who are doing that kind of work. And this is like what I call working smart. This is using information that's out there in the world and bringing it in a way where you can make more strategic sense out of it in the way that patients are treated. So here's an example that I'm showing in this slide about how they looked at studies that had been done with different kinds of lasers. For diabetic retinopathy, and what they were able to show was that there was no one type of laser that was superior to the other. And then finally, the most recent addition to our department is this professor, Jayashree Kalpathy-Kramer, who is a specialist in artificial intelligence and machine learning. And what Dr. Kramer is able to do is to develop technology to look at images that are collected in the ophthalmology clinic. So these are retina images. And to look for features in these images that may give some clue as to the outcome of that patient's response to therapy. And so many times doctors, they need as much diagnostic information as they can gather, but it's limited to the power of the diagnostic tool. What artificial intelligence is able to do is to look for patterns in the images that are collected from the retina scanning and to find patterns in these images that predict the outcome of that patient's therapy. And they find patterns in the images that a human doesn't have the capability of recognizing. Let me give you an example. Artificial intelligence is able to scan all of these images, and there's something in the image that they can see that tells it if the patient is a male or a female. And doctors, when they look at the images, they have no idea. There's just no clue. But this artificial intelligence method with machine learning is able to detect whatever subtle changes there are, differences between male and female, and can tell the doctor what sex it is. That's really amazing. So they look for these kinds of features in these images that give some idea of how this patient is going to respond to therapy. Artificial intelligence. So as a result of our growth and our basic research, we've seen a growth in the amount of our success in getting funding for research. Research is very expensive. The main source of external funding for our research is the National Institutes of Health. That's a federal agency. Right now, well, in last year, our annual funding was up to about $3.5 million. This year, our funding will be in the $5 million range per year. So it's a nice increase over the years. What do I see as our challenges to the success of our research program? Funding is going to be a major challenge. It always is. I mean, the cost of doing research is high. The cost of personnel is going up. And the fuel that feeds all of that is research funding, primarily through the National Institutes of Health. And it's a competitive process. We apply for grants. We have to compete with a lot of other scientists who are after the same pot of money. The success rate for NIH grants is somewhere in the 15% range. So we're quite successful at getting grants, but it's not easy. And so that's a big challenge for us, is to keep the funding stream going. One of the other big challenges we have is workforce talent. Denver is becoming a better and better location for science talent because of the technology that's being developed between Anschutz and the Boulder campuses. And a lot of biotech companies are being established in Colorado. But there's not enough talent locally to fill the jobs that we have available. In fact, we just had a big hiring phase this past year and had to go all over the world to find people to bring to Denver to work in our labs. So we need to fill that pipeline. This is what I was mentioning before about the importance of establishing the next generation of scientists. We need to create a pipeline to feed the laboratories that need the scientific talent. The pay is pretty good. And so it's a very attractive working environment. The challenges, the gratification of doing good science is really high. The work environment is really good. It's just that we're competing with a lot of other outfits for the same talent pool. So this is going to be a challenge for us. One of the other challenges that we face is the high cost of doing this translational research. You know, it's expensive to do the basic research to come up with new therapies. It's incredibly expensive to take that to the clinic. And so one of the things that we try to do is to either create companies that can raise capital to be able to do clinical trials, or to license out our technology to companies who themselves can raise money to fund clinical trials. But it's a really difficult process to get through, just to overcome the cost of getting your material into the clinic. And then something we all face is inflation, the accelerated cost of doing research. One of the issues that we face, you know, I was mentioning that federal funding is one of the major sources of our research funding. The cap to research grants has not been increased in 20 years. And so you can only ask for so much money, and they will only award so much money. And yet the cost of doing research is going up, even though the funding level is not. And so we have to do our own fundraising here locally. We depend on philanthropy. We, we have grateful patients who help us out with some of our research expenses. But we just simply cannot rely on federal funding to fully cover the cost of our research. It's just, the buying power of research grants has gone down pretty dramatically. And one of the other things that we always need is ways to modernize our infrastructure. One of the things, laboratory equipment is expensive. One of the things that we currently find ourselves needing is an upgrade to our gel imaging system. This is something that costs in the $70,000 range. This is a piece of equipment that is shared by all of the investigators in the department. So all the people you saw pictures there, they have people who use this gel imaging system. So it's a really important core piece of our research infrastructure. We also have need for instruments that can handle very small volumes of biological reagents. And we do this because we need to economize on how we do our experiments, and by miniaturizing all of our experiments into really micro volumes, we can do more experiments for the same amount of money. But you need to be able to handle these small volumes of liquid. And so this liquid handling equipment enables us to do that. This is, again, something that's shared across the department by many laboratories. And then, and these liquid handling devices cost in the $25,000 range. And then we have a constant need to upgrade our computer systems. All of the instruments we use in the laboratory are computer-driven. We're constantly going through operating system upgrades and things like that, and software upgrades. And so we need to be current with our hardware, computer hardware. And so we typically go through about $10,000 a year on that. Okay, so I hope this gives you a flavor for the variety of research that we're doing, the strategic focus that we have on developing therapies that we can bring to the clinic, and our focus on making an impact in our research.
Tell us some of the successes.
On the basic research side,
Well, that you've been able to bring to the market.
well, we don't have any. Well, we have one device that has been brought to the market, a glaucoma surgical device that was developed by Dr. Cahook. We have several technologies that are still in development, and that one being a therapy for glaucoma. So it's a neuroprotective agent for glaucoma. We have a set of molecules that show promise as anti-cataract drugs. We have a set of molecules that show promise in the diabetic retinopathy. But these take years and years and years to develop, to get corporate partnerships so that these can be brought to the clinic.
Wow. What about dry eyes? I have a couple of friends who have problems with dry eyes, and I had never heard of the
Yeah.
difficulties that a lot of people have.
Yeah, we do not have a dry eye program, a research program. Yeah, I recognize it's a big problem. Yeah,
Good to think about.
yeah, thanks.
When we visited you with Dr. Choi, you had just hosted a conference here, and everybody was really pumped about a couple areas in your research where you appear to be ahead of the rest of the pack. And not being a scientist, well, I didn't track completely with what all that was. I just remember how enthused everybody was, and that you have a young man that's in your group that's on top of things. If you can say anything about what he's doing.
Sure. The occasion for that visit was that ARVO, the Association for Research in Vision and Ophthalmology, had just had its annual meeting in Denver. And so all of the world's leading scientists were here at that time. And we had several who came to visit the department after the annual meeting. And one being the the director of the National Eye Institute, who came and gave a talk and visited our laboratories. One of the areas that we're particularly excited about is our cell site program. And one of the scientists in the cell site program is Miguel Flores, who's sitting in the back of the room here, who had just made a big discovery about some of the mechanism of macular degeneration and some of the molecular mechanisms where different cells in the retina communicate with each other. And so Miguel gave a paper at the ARVO annual meeting and released some of these results to a lot of excitement. And so he's our rising star.
Didn't see you coming in. Welcome.
Thank you. Sure, yeah.
Are you doing some research into the pediatric side? When I have a family whose children are going blind, and do you have some pediatric investigations?
We do have a pediatric, so this is at Children's Hospital, doing a lot of research on strabismus, and this is the process where the eye alignment gets out of whack, and trying to understand how the muscles can be retrained to keep those eyes in proper alignment. That's the major area of research in our pediatric ophthalmology program.
One of the things you said suggested that you have you have researchers and you have clinicians. Are there people who do both?
There are a few clinician scientists who see patients and also maintain an active laboratory program. So Dr. Pencheva, the lady who's in our glaucoma program, has an active clinician scientist program. Those are relatively rare, just because the clinical demands for someone who can see patients are so high that it's hard for them to peel off enough time to come to the laboratory and direct the laboratory research team. So that's why this teamwork arrangement is so important.
Do researchers tend to start out as clinicians and then switch, or the other way around? Or?
Rarely.
Okay.
So basic researchers tend to be laboratory focused, without medical training and without the sort of credentials that you need to be able to see patients.
Okay. Now you talked also about the collaboration. If I am a clinician and I am trying to see if it works in practice, am I somehow in the middle as I'm doing that? Or?
Exactly. Yeah, you're as a clinician will meet with the basic scientists, will help design studies to test new ideas that could be brought to the clinic eventually. But the basic science needs to be done first. Very often, a new therapeutic concept will be tested in animals. And so the clinician can be part of that process of treating animals as they would a patient. And the basic scientist often doesn't have that skill set. So the clinician comes to the laboratory and helps to work with the animals to establish the concept. The therapeutic concept is solid that could then be taken to the clinic in the future.
Let's suppose that somebody drops a couple $1 million from their helicopter or whatever, and somebody says, "Oh, this would be good money to use for." Sorry. Is that better directed towards a researcher or towards a clinician, or the new? I mean, not the fellowship. Let me say an endowed professorship. Let's put it that way.
Yeah. Well, I would say the biggest impact would be for a basic scientist would benefit from an endowed chair such as that, because what that does is it provides the steady funding to be able to pursue high risk, high reward research without without having to structure it so that it's safe research. Some of the biggest, highest impact research are novel ideas that seem like out-of-the-box thinking, that once they're pursued and once they're studied carefully, can lead to very innovative new therapies.
Now, if I ask that question of a clinician, would they respond the same way?
I think so.
Okay.
I think so. And because this is how big breakthroughs are made. This is how, who would have thought that culturing stem cells in a petri dish would lead to growth of an eye, of a retina? I mean, it was like unheard of. And yet someone had to have the courage to try it. And someone had to do the planning to enhance the chances of success, but it's still very high risk. And this is the kind of research that benefits from an endowment to pay for the high risk, high reward research.
There was recently an announcement of a large endowment, I guess, by Gates in regenerative. And could you tell us a little bit about that?
Yeah. So the Charles C. Gates Foundation started at the Gates Center for Regenerative Medicine back in 2006. And that center created an infrastructure for doing stem cell-based research. And it reached a certain level. There was a critical mass of faculty who had been recruited and who were working together in the area of stem cell research. The leaders of the Gates Foundation recognized that once that seed had been planted, really the next quantum leap needed to occur to really make this center successful. And that quantum leap requires funding, requires support, financial support. It required sort of a reorganization of the leadership. And so the leaders of the Gates Foundation came to the university and said, we would like to propose to help fund that quantum leap. And we will commit $100 million to get the Gates Center to that next level. And the university agreed to match that with $100 million. So there's a $200 million war chest now that's been committed to help take stem cell-based research in regenerative medicine to that next level. What is that next level? That next level is to be able to take this technology like our retinal stem cell transplant through the very expensive process of FDA licensing, through all of the regulatory steps that have to be taken for showing that you can produce the retinal tissue in a manner that's safe to put into patients. Everything we have done until now is research-based in animals. To be able to grow that retinal transplant in a manner that's suitable for a human patient is very expensive and has to go through a tremendous amount of testing. There's no way that ophthalmology could afford to do that. But with $200 million, that can help cover those costs. Now, all of that $200 million is not being set aside for the retina transplant. The retina transplant will be part of that. But to answer your question, the Gates people came up, they initiated this concept to put in $100 million, and the university matched it with $100 million. A new institute director was named. It's Terry Fry. He's a molecular oncologist who uses stem cell-like technology for cancer therapy. Part of this infrastructure that was created by the Gates Center, which will benefit from this $200 million, is a biomanufacturing facility where we can grow cells and tissues in an environment
that's acceptable to put into patients. And so this $200 million helps to cover some of the costs of operating this biomanufacturing facility. But it's an amazing gift. And it's transformative because the investment that's been made by ophthalmology, by the Gates Foundation, by other entities within the CU Anschutz campus community has enabled the university to grow from having no stem cell program to having a program that has produced material that's now ready to go through the FDA regulatory process. That's amazing in a relatively short period of time. But to go from where we are now through FDA approval is extremely expensive. And we need to be in the driver's seat of that. If we tried to license it out to a drug company or something like that, we would lose control of it. And we would not benefit from the investments we've made in bringing that technology to where they are now.
So your plan is when this gets approved by the FDAs, there will be some financial return back into the future.
We hope so. Yeah. We're not counting our chickens, let's put it that way. But this is, how future research can be financed is for the university to get a return on some of the investment that they've made in hiring faculty, building out a medical campus like you're on right now. It's only fair.
When you talk about developing this retina with a stem cell, do you see it going? How do you see it going into someone's eye? I mean, is it going to be done outside and then transplanted, or can it be grown inside?
Yeah, great question. It's grown in a petri dish.
Right
So
now?
it's grown in a laboratory, and it will be, well, as far as we know, it takes about 6 months of growth in the laboratory, starting with the stem cell, to have a retinal patch that's suitable for transplant. And these are under very tightly controlled conditions. We have special secret sauce that gets put on the cells when they're growing and developing. There's a very precise timing and the different mixtures of materials that get put on these cells. It's all been worked out very well. And so that could not be controlled in the eye. It's something that's laboratory controlled. And it takes about 6 months. What we're hoping, this is like pie in the sky. What we're hoping is that we will be able to develop a retinal transplant from what we call a universal donor cell. So that if we had cells that had been treated in a way that make them compatible with multiple different recipients, rather than someone who's receiving their own cells, then that would make it much easier to scale this technology. Because as it is now, if I was going to make one for myself, I'd have to donate cells. It'd have to grow in the laboratory for 6 months, and then it would possibly go to my eye. So that's a long waiting period, and it's extremely expensive. If we had an off-the-shelf therapy that would be universally acceptable to many different patients, not everyone necessarily, but many different patients, then we could treat. It would be much less expensive and much more efficient to treat patients. So we have collaborations with groups that are developing universal cells that we'll be testing if we can grow retinal patches from those cells.
Where do you get universal cells?
I'm not really sure where they start with, but they get genetically modified.
Oh, okay.
Yeah. There are certain tissue types, across the world that can be put into a relatively small number of buckets. And many different people can have some compatibility with cells from one bucket. And so that one bucket couldn't treat everyone, but it can treat a large segment of the population.
It's like blood type.
It's exactly like blood type. So instead of ABO, it's a different set of molecules that are recognized by the immune system. And it's possible to genetically modify these cells so they don't make those molecules that are part of the rejection process. So if they can take those away, then the cells would be less likely to be rejected.
Wow, you guys have come a long way.
So to kind of made me think about with her question is, do I remember correctly that the ultimate goal would be that the local ophthalmologist would be able to do the transplant?
Oh,
Boy, that's a long way off. I mean, ultimately, we want to make it available to everyone. Whether it's something that your local, I would say no. I mean, this would be like a heart transplant or a lung transplant.
Limbs crafters.
It's a very complex process to get the retinal tissue in there. Miguel, did you have a?
Yeah, I was thinking about that. You always have really wonderful questions, really good ones. So we developed a system where we can prepare the transplant. We, as Dr. Petrash mentioned, we developed new surgical tools. But also we were trying to make sure that we can make the transplant in the lab, and then we can ship this over the world. So we did a career to put this transplant within the career in a specific media to send it to Baltimore or England or Spain or whatever place in the world. We have the surgical tool for this transplant to be able to clean the eye. And we have already measured all of this and make sure that everything is healthy once arrived to the hospital. But I think the critical part Mark is mentioning is to do the training with the doctor. We have here in the department, Dr. Matthias, who is working on that for the last 4 years, like surgical tools and all the equipment, and we need to do transplantation. But in the future, I mean, it's something that we hope to be able to do. We have the tools ready, we have the transplant ready, and we will be able to see this over the world. But the hard part will be training new doctors to know how to use the stem cells and the technique to use the surgical tool. But it's something that we think it's feasible to achieve in the long term.
I would think so, because, you know, the eye bank is cornea, and it must have taken a while to train people to be able to take that cornea
Yeah,
and put it. So it would be.
yeah.
Same.
I mean, it's a matter of many different trainings involved on that. But it's true that we are kind of the factory of the transplant, and we have everything to ship, like the tools. That when we prepare this, and we work here in the facility, when we prepare the transplant, we are able to put in a specific recipient like 10 transplants, even though the doctors are going to use 2. We ship them by a carrier like FedEx to us to see how it works and how safe it is after going through FedEx around the country and go back. And it was safe, we opened, it was not contaminated. We are able to show Dr. Matthias here how to get the transplant, and he, with his skills as a doctor, a retina surgeon, he's able to make it happen. But the critical part will be to do the training, but it's what happened in the school, right? On medicine, they have to be training for everything. So we are thinking about something really far away because it's not ready yet. So doctors are gonna be, they're gonna have that time to learn how to do that, but I think it's really something helpful to achieve.
Mark, very informative, thank you.
Thank you.
And you know, I met you a while ago, it got down to us as lay people. We understood what you were explaining.
Thank you.
We appreciate that. So our next thing on the agenda is a break, and then we're gonna meet up for a tour. And I understand you bring a face mask with you, depending on where we're going, you'll need that. So take your break, and then we will meet there and form our groups and go.
Where are we meeting,
So-
area, Mr.
John?
Here,
We
okay.
can take you to the research area for the tour, so whenever you're ready from your break, we'll just walk over from here.
Who is that
If
person?
we need to use the restroom,
The agenda showed that some of us would stay here, some over there. We're all gonna go over there, and then we'll divide into our two groups there, is my understanding.
Right. Right.
And then we'll come back here to tour this way.
What time do you need to be back here? Is it firm?
We should be back here about 3 o'clock, about an hour.
We need time so we can tour this building before they close down.
Okay.
We figured by you being able to talk here to everybody,